Thank you very much. I very much appreciate the kind invitation. It's a great privilege and an honor to speak at this conference today and to share some of our work. So I want to thank the organizers for having us today. The title of my presentation today is AP-4-associated hereditary spastic paraplegia, a multimodal strategy to finding a cure for an ultra-rare disease. And I'm very excited that I can share some work on SPG47, SPG50, SPG51, and SPG52 that we've been doing over the last 3 years. So I hope you can see my screen in just a moment. Let me see. Okay. So with this, I would like to get started. And first acknowledge the fact that a lot of the work that I'll be presenting today was made possible through a very generous grant from the SPF, from the Spastic Paraplegia Foundation, that really helped us get things off the ground. So I'm very grateful for that. So how did this all start? Our research program into AP-4-HSP started about 3 years ago, and it really started with a chance encounter. It started with two patients. These are two of my patients, Robbie and Molly, who we met about 4 years ago, and who at the time were only the 9th and 10th patient to be diagnosed with SPG47. So while we were able to make the diagnosis through genetic testing, we very quickly realized that there was very little research being done on SPG47. And much of the work that I'm going to show you on the development of a research program was really spearheaded by these two families, because they got together and they created a foundation called the Cure AP4 Foundation. And that really sparked our research program, really gave us a very good partner to work with. And as a first step, since we're at Boston Children's Hospital, we're in a unique position to see and to research rare diseases. We very quickly realized that there were more patients out there. So we found a set of patients in North America, and we put together a first report describing these North American patients for the first time. But it was only a small step. We very quickly realized that we had to do more. And that's what I'm going to highlight today. I'm going to talk about three things, essentially. First, I want to talk a little bit about challenges of studying an ultra-rare disease, but also opportunities. In the second part, I would like to show you how we approach and understanding the clinical disease spectrum, how we try to define the disease spectrum, and how we try to set the stage for clinical trials. And then in the third part, I would like to share some insights into key molecular mechanisms that we think are at play, and how we can use these insights to develop a screening program, a screening campaign that tries to find new therapies. And I'm preaching to the choir here, but there are some unique challenges in studying ultra-rare diseases. And I think it's sometimes important to remind yourself of that. One key fact that I always struggle with is the fact that, although by definition rare diseases affect only a small number of patients in any given disease, in aggregate, they affect over 350 million people worldwide. And what struck me is that of these sort of 7,000 rare disorders that we know of, very few have specific treatments. And this is shown on the right side on the graph. While our understanding and our ability to diagnose rare disorders has really taken off in recent years with the advent of genetic testing and the availability of genetic testing, and our understanding is increasing, this is really, the development of treatment has really lagged behind this trend. So you can see here in the blue line shows you the number of publications over the years, and then the red and green line show you the number of approved therapies. And while there is a positive trend, I would say, there's really a big gap between understanding disorder and finding a treatment. And one of those factors that contribute to this gap is the fact that for many of these rare disorders, we don't have adequate disease models and reagents to study the disease in the laboratory. And on the clinical side, there's often a lack of detailed phenotyping and a natural history study that would allow us to conduct clinical trials in an appropriate setting. And I think at the root of all of this is the problem that for any rare disease, funding is limited. And until recently, although there are more and more incentives now, commercial interest in developing therapies was limited. So that's the framework that we operate in. Now I would like to talk a little bit about the specific disorders that we study. And I know this audience is very familiar with the terminology, but I want to show you what I mean by AP-4-HSP, or adaptive protein complex 4 associated HSP, using this very simple cartoon here. So the AP4, or adaptive protein complex 4, is a protein complex that consists of 4 different subunits, shown here in the center of the slide. And any of these 4 subunits is encoded by a different gene. And these genes are very different. They're located on different chromosomes. They look very different. But in order to have a functional protein complex, you need to have all 4 genes present, and you need to have all 4 proteins present. So no matter if you have a loss of function variant in SPG47 or SPG50, 51, or 52, the result is the same. If you lose one of the subunits, you lose function of the protein complex, and the molecular consequence, and hence the clinical problems, are similar. So we can think about this as one disorder. And just to illustrate or to highlight that point, I'm going to show you 3 videos of patients of mine. They're roughly the same age, and as you can see on the bottom, they have different mutations, including in different subunits of the protein complex. But what we will appreciate is that they share a lot of similarities. So I'm going to show you the videos now, and you see that all 3 walk with have difficulties with gait, and they walk with a somewhat broad-based spastic ataxic gait. All 3 of them also have a low muscle tone in their trunk. And you can't see this on this video, but all 3 have significant delays in their development, both motor and speech. So this shows you that although the subunits that are affected are different, the disease that results from that is similar. So when we set out to start our research program, we identified 2 goals that we felt were necessary to achieve before we can move into a clinical trial. The first was to really improve our understanding of the disorder, and we sought out to achieve that by creating an international registry. And the second was to create a natural history study, a longitudinal study, that would tell us what areas are affected, what symptoms are present, and how they change over time, so that if we have an intervention that we can test in a clinical trial, that we could look at this as an outcome. So first, about the registry, we sought out to develop a cross-sectional analysis, which would tell us about the disease spectrum, would define the patient population, and help us look at disease manifestations across the age spectrum, which is hugely important information in order to guide symptomatic treatment, but also in order to be able to counsel families, really. And we decided to do a first cross-sectional analysis, meaning an analysis of all cases we could identify, to be able to look at markers or endpoints that we could then follow up longitudinally in a natural history study. And the goal of the natural history study really is to track disease progression in a standardized way and to define endpoints for clinical trials. So this is some data. In the beginning, I mentioned our first 2 patients were number 8 and 9 in the world to be diagnosed with SPG47. And looking at all 4 subtypes, we now know of about 200 patients. This is data from December of last year, and since then, the numbers have gone up to 200 patients. And you can see they come from different countries, different areas of the world. You see the majority are from Europe or the Middle East, and there's a subgroup that is located in North America. So in our registry, in our cross-sectional analysis, we rely on a standardized questionnaire, and we designed this very broadly because we wanted to be able to capture any disease manifestations or problems in a very broad way. So this is a questionnaire that we asked local clinicians to complete. It contains 169 items. It's designed for a cross-sectional analysis. It can be completed in about 20 to 30 minutes. And it includes a number of rating scales that are commonly employed, such as the Spastic Paraplegia rating scale for children and young adults who are ambulatory, as well as other scales that look at motor function or quality of life. And as part of this study, we also collect any MRI scans that might be available from clinical testing. So here's some data that we found looking at the first 156 patients enrolled in the registry. We first found that two subtypes, SPG47 or AP4 beta 1 and SPG50 or AP4M1 are the most common subtypes, each accounting for about 30 to 40% of the population. We also found that the age spectrum was wide, but most of the patients that were diagnosed in the last 3 years were under the age of 5. And this is because genetic testing is just more available now as it was years ago. We also found that despite the fact that we're diagnosing kids at a younger age, on average, there was a great delay in diagnosis. And this is really an important point for us as clinicians. I think there's a lot of room for improvement. On average, a patient becomes symptomatic in the first year of life, as I'll show in the next couple of slides. But it takes on average 9 or 10 years for the diagnosis to be made. So there's a big gap there. And I think we need to close that gap. The most common diagnosis that was made in cases that achieved the diagnosis late was cerebral palsy. So that is an important mimic of this condition, and I think an important factor for us as clinicians to recognize and to realize. So through our very detailed analysis, we were able to define a couple of things that weren't described in the literature before. We found that a subset of patients had a short stature, and the vast majority of patients had a small head size, something that we call microcephaly. We also found that foot deformities, as shown here on the right-hand side, were relatively common. The main symptom that brought these children to the attention of a neurologist or geneticist was developmental delay. And shown here in a somewhat complicated graph are common motor milestones that we assess as pediatric neurologists. And you can see that on the top here, you can see the age at which these are achieved. So for example, sitting is typically achieved at 6 months. However, in this patient population, sitting is achieved late at around 16 months. And you can also see that unassisted walking, so walking without any significant assistance, was only achieved by about 40% of the participants in this study. So this gives us an idea of the developmental trajectory of these patients, and developmental delay is universal in this condition. Now, obviously, the defining neurological symptom of HSP is spasticity. And in this patient population, in children with AP-4-HSP, we found a general pattern. We found that there's often low muscle tone in the neonatal period and in infancy, but over time, spasticity begins and typically involves the legs first. And in a subset of patients, that progresses to involve the arms as well, leading to some complications, including to difficulty swallowing, for example. For us as neurologists, we were also, we did find, and this is important for us, that it's not only a condition that presents with spasticity, but also with extrapyramidal movement disorders, including dystonia and ataxia, at least in this subset of patients. One important symptom that we discovered was that about 2/3 of patients presented with seizures. And this was often initially in a setting of fever. As you can see here, about 60% had what we call febrile seizures, and about 66% qualified for a diagnosis of epilepsy. And this is important because we found that seizures tend to develop earlier than spasticity. So oftentimes these patients will present at the age of 1 or the age of 2 with seizures. And in our practice, in a pediatric neurology practice, seizures are recognized as part of many genetic conditions, and genetic testing for epilepsy is advancing rapidly. However, these genes, AP4 beta 1, M1, epsilon 1, and S1, are not included in the gene panels commonly used to detect genetic epilepsy. So we're trying to change that. We also found that a subset of patients had medically refractory epilepsy. And in these patients, we found on MRI scans that certain brain malformations were more common. We had a chance to look at about 100 MRI scans of patients, and we discovered a common signature. We found that the corpus callosum, which is the connection between the two hemispheres of the brain, was thin, particularly here in the back part of the corpus callosum. And we found that the fluid-filled chambers called ventricles were enlarged. And we think this is due to a loss of white matter in this region. We did find that in a subset of patients, there was evidence of brain shrinkage, both of the hemispheres as well as the part of the brain called the cerebellum. And in a subset of patients, we found distinct brain malformations that can predispose to epilepsy, such as polymicrogyria. On the molecular side, and I'm not going to go through very much detail here, but on the molecular side, we discovered that the mutations mapped to many different axons of each of these genes. About 80% of patients had a homozygous mutation, about 20% had a compound heterozygous mutation, and the vast majority of variants are frame shifting or nonsense variants. But there's a subset of patients who has missense or splice site variants. Now, what did we learn about disease progression, or what did we learn about disease severity across the age? And this is what we tried to get at using multiple rating scales. Shown here are the data for the spastic paraplegia rating scale, and you can see that there's a moderate trend towards higher scores in older patients as a surrogate of disease duration. We also found that disability increased over time. We did not find any differences between the four subtypes in terms of disease severity. What we did find, however, was that children who presented with epilepsy, and again, this is something that happens very early on, tended to have more severe motor features later into the disease, which is important for us because it gives us a marker to look at to stratify and see who might develop motor complications more rapidly. We also learned that some of the rating scales that we used were not useful, and this was an experience that we anticipated. As you can see here, for example, these two rating scales had a clear ceiling effect, with the majority of probands reaching a high score. So this does not give us the necessary capacity to stratify disease severity. So in summary, from this first effort, we were able to define the disease spectrum. And this is hugely important because it gives us as clinicians the opportunity to recognize the disorder. It raises awareness about the disorder. It helps us anticipate problems and counsel families. And it also helps us design a study that looks at these problems over time to be able to then say, if we make an intervention, how does that change over time? And that is very important. So listed here are the core features that define AP4-associated forms of hereditary spastic plegia, which includes developmental delay, hypotonia, spasticity, microcephaly or small head size, seizures, and then certain radiographic findings. Now, with this data set in mind, we then set out to design a longitudinal study, which is currently ongoing. And in this effort, we're trying to use standardized clinical assessments. We're trying to develop biomarkers, which is important for monitoring response to therapy. And then also to define endpoints for interventional trials. And as you might be able to imagine, there's some unique challenges when we design a study like this for an ultra-rare disorder. In our case, we felt that one big challenge, and this is increasingly relevant in a world that is facing a pandemic is the fact that our patients are, well, first of all, it's an ultra-rare disease, but they're also geographically located in very different parts of the world, which makes it sometimes difficult. We recognize that the disease manifestations were heterogeneous and that the natural history would probably reflect that heterogeneity. We, as pediatric neurologists, are very familiar with the concept that a brain disorder unfolds or evolves into a time period where there's a lot of brain development happening. So that makes it difficult to measure certain things. And then lastly, many of the tools that we use in pediatric neurology to assess behavioral or cognitive function depend on motor function. So we cannot use them really in a child who has problems walking or problems manipulating objects. So when I think about AP-4-HSP, I think to me the concept emerges that there's both a neurodevelopmental disorder as well as a neurodegenerative disorder, and this makes it challenging to study this disease. Now, how do we go about the natural history study? We designed a follow-up questionnaire, and we focus on those areas that we think are likely to change over time. Again, we designed it in a way that would make it feasible to complete this during a routine office visit. We designed it for longitudinal analysis. We made sure we can use it in all age groups. And as I said, we focus on variables that are likely to change over time. And as I said before, we try to include established rating scales as well, including measures of quality of health, health-related quality of life, which is important because we want to know what families think are important areas. So that is sort of the clinical part, and this work is all to set us up for the day when we have a therapy in a clinical trial. And now I want to spend some time talking about what we do in terms of a translational basic science research program. And this is a concerted effort with many collaborators. We at Boston Children's Hospital focused on two areas. We decided that we needed to develop disease models, and we needed to develop reagents and tools for the community. And secondly, we decided that we would take on a small molecule screen and look for therapies through a screen of thousands of compounds in patient-derived cells. So to achieve this agenda, we set out to collect patient cells. We developed what is called, what are called induced pluripotent stem cells. And we use these induced pluripotent stem cells to make nerve cells, which give us a unique tool to study a predominant brain disorder. And then we set out to develop some specific reagents without which no research is possible, such as antibodies. And we also set out to develop a zebrafish model, which I'm not going to talk about today, which I think will be important in our efforts to find therapies. So for the small molecule screen, we had to establish a primary screening assay, some additional assays called orthogonal assays. We developed neuron-specific assays because AP-4-HSP is a brain disorder. And we set out to use these tools to screen both FDA-approved compounds as well as novel compounds. That's the general approach that we take. So I'm coming back to this slide, and again, to remind us that there's a big gap between our knowledge and the translation into therapies. And when we think about modalities of therapies for rare disorders, several modalities come to mind. And this is nicely summarized in this review paper. The most traditional way of developing therapies is to search for a small molecule. But then there are also newer avenues of therapy, including genetic therapies such as antisense oligonucleotides or gene therapy or gene replacement therapy. To orient everyone, with AP-4-HSP, we're in this category. We think we have a loss of protein function because of mutations on both alleles. And for this kind of disorder, several approaches are possible in the right circumstances. The one that we focus on is that of small molecules. I want to talk a little bit more about that. So generally speaking, if we look at the therapies that were approved in recent years, small molecules make up the majority. And that is because there's been a lot of experience with this approach in the pharmaceutical industry as well as in academia. So it's a very traditional approach, has several advantages. But again, to orient everyone, out of the 3,000 proteins that we know are disease associated, only 700 have an approved drug that targets that protein. So there's a lot of opportunity to identify additional therapies. That identification of small molecules relies on screening of cell lines with libraries of compounds. And in the last couple of years, the advent and the availability of better cell models, such as iPS cell-derived models, has really enhanced the translational relevance of these models, because most of these screenings traditionally are done in cancer cell lines, but a cancer cell might behave very differently from a nerve cell. And another approach, and this is something that we're trying to develop, is a multimodal approach, meaning we test therapies not just in a cell line, in a stable cell line, but we try to use patient-derived cells in addition to model organisms such as zebrafish. And one advantage is that drug repurposing is possible. It's possible to identify a compound that has established safety profiles, that has established pharmacodynamic and pharmacokinetic profiles. So that is an advantage. And just to highlight two examples of a success of this kind of approach, there's a drug that was approved for certain types of cystic fibrosis that was identified through one of these screens in the cell line. And the same is true for repurposing. There were medications that were used in the past for different indications that were found to work in other disorders through a screen like this, not through clinical serendipity or clinical use or any specific reason, but really through a concerted effort in screenings. So again, that's the framework that we operate in. And now I would like to spend a little bit of time talking about the cell biology of AP4 and the cell biology of AP4-associated hereditary spastic paraplegia. And I understand that this is very technical, but I want to highlight a couple of things. AP4 as a protein has a very important role in the cell. It is important for the trafficking of other proteins inside the cell. So the vast majority of proteins are transmembrane proteins, are trafficked through a part of the cell called the trans-Golgi network. And this is where AP4 sits and helps package these membranes into vesicles that then go on to different parts of the cell. So it's kind of like the post office. It kind of sends it to different places in the cell. And that is very important because any of these proteins needs to end up in the right location. We know that AP4 is expressed in the nervous system. So I'm showing you here data from both rodent experiments, but also from expression analyses in the human brain. And we know that it is expressed in the brain. And when we first started our research, we were in a fortunate position because at that time, 3 independent groups had identified the cargo that AP4 helps traffic, so to speak, the mail that AP4 helps to deliver. And that was mainly one protein, to our surprise, which is called ATG9A, which is an important protein in a pathway called the autophagy pathway. And I'm showing one key figure here from our from the work from our collaborators, Alex Davies and colleagues in Cambridge, UK, where they used mass spectrometry-based methods to identify these cargo proteins, and the ATG9A stood out. Importantly, what these groups identified was that ATG9A in cells that lack AP4 gets trapped in the trans-Golgi network, and this was a very strong phenotype. Present in cells, but also present in mouse tissue of knockout mice. So these are nerve cells in the cortex of these knockout mice, and you can see here, if you look at these pictures here, that ATG9, this is the dark signal, accumulates in the region around the cell nucleus. So from this work, the following hypothesis or the following working model emerges. We think that mutations in AP4 lead to a loss of AP4, which leads to a sequestration of ATG9A in the trans-Golgi network, And if you lose ATG9A from parts of the nerves called the axons, you get axonal dysfunction and degeneration over time. So we think that is the molecular cascade that happens in AP-4-HSP. And this is supported by circumstantial evidence in mice in the sense that if you knock out ATG9A, you essentially get the same phenotype that you get if you knock out AP4. And that tells us that these two are linked and part of the same pathway. So in the first set of experiments, we set out to test whether ATG9A was mistrafficked in patient-derived cells. So we used skin cells from patients, as you can see here, from 3 patients with SPG47 with AP4 beta-1 mutations. And we found that in lysates of these cells, ATG9A was robustly increased, as you can see here. And that indicates to us that the trafficking or the turnover of ATG9 is perturbed, very similar to what the other groups had found in other cell models as well as in the mouse model. We then looked more carefully and we found that ATG9 accumulated in the paranuclear area. You can see that here, in this sort of white signal around the cell nucleus. And this was a very robust finding. We found this in every cell line tested, and we tested a number of cell lines. We found it in each one. And this is important because when we saw this, we recognized, this is a very good marker that we could use to test therapies against. This is a functional readout that tells us whether AP4 is functioning or not. And secondly, it's also a marker that helps us make a diagnosis in cases where the genetics are not as clear. So for example, where you have a mutation that you don't know what the impact of it is, you can use this assay in cells to really look at the function of the complex and to help make a diagnosis. This is the quantification of the experiment I just showed you. And this is again looking at protein levels where we found that ATG9 is increased across the board. We then went on to prove that this increase in ATG9 in the trans-Golgi network in the paranuclear area was due to a lack of AP4. So we did essentially what gene therapy would do in a clinical trial. We re-expressed the protein. In this case, in the Petri dish, we used a lentivirus. We re-expressed the missing subunit, and we found that that decreases ATG9 level to levels that are close to controls. So that proves that all of this is secondary to a loss of AP4. Now, all of what I've shown you so far was in skin cells, in non-neuronal cells. So the next step, we asked whether the same was present in nerve cells derived from patients with AP-4-HSP. And to do this, we took the approach of first generating induced pluripotent stem cells and then differentiating these stem cells into nerve cells using overexpression of certain transcription factors. And these, what we call excitatory cortical neurons, were then subjected to the same experiment where we look at ATG9 in the cell. And we found that similar to the fibroblasts, we found that ATG9A, as you can see here in this pseudo-colored picture, accumulated in these cells. And this is quantified here. That was important for us because it proved to us that the phenotype, this molecular finding, was preserved between skin cells and nerve cells, which is important because that means we can use the skin cells to do our screening because they're much easier to handle, they're cheaper, they're faster, and then use the neurons to verify and to validate any findings in the right and relevant context. So that's what we set out to do. We also discovered that these nerve cells had a problem with growing neurites, growing axons and dendrites, as you can see here. So that is very similar to what people discovered in the mouse model. So then we took this phenotype and we asked, can we develop this into a screening assay? And the very simple thing that we're trying to achieve here is really to make a cell that looks like this, which is a cell derived from one of our patients, look like one of the control cells like this. So we're looking for any molecule that can change this picture into this picture. That's the very simple approach that we take. And we were able to scale this up to a format where we can use 384 weld plates with thousands of cells, and in each experiment we can screen thousands of cells, and that gives us a lot of statistical power. So this is the general approach. We developed an automated pipeline that uses high-throughput confocal microscopy to look at the localization of ATG9 in a trans-Golgi network and in the cytoplasm. And we're asking the simple question whether a compound can change the localization pretty much the same way re-expressing the missing gene would. So having established this pipeline, and I'm skipping over a lot of work, I'm skipping over a year of optimizing this automated pipeline that largely relies on robots and automated microscopy now, to show you one of the first proof of principle experiments. And at Harvard, we're fortunate to have access to a number of compound libraries through our ICCB. And we decided to use a first library to test our pipeline. And we chose a library that is optimized for brain disorders. It's called the NINDS2 library, which is a library of 1,040 compounds that have known brain activity. About 75% or so are FDA approved. In this first screening, we screened a single concentration and duplicates. We used a single cell line from a patient with SPG47. We had predefined thresholds for assay metrics, predefined thresholds for active compounds, and predefined threshold for toxicity. And just to give you a snapshot of the results, this is the distribution of z-scores, so the degree of change in ATG9 ratio. And we're looking for compounds that can change this ratio by at least 3 standard deviations. And in this library, we found 59 compounds that achieved this predefined threshold. When we looked at assay metrics, we saw that the plates of the assay perform very robustly. So here we're looking for the light blue bars, we're looking for a number greater than 5. And for the red bars, we're looking for a number greater than 3. And all of the plates achieved this assay metric. And then looking at our compounds and how well they perform, this is a very complicated graph, but I just want to highlight the fact that we're looking for compounds that move from the dark blue line, which are patient cells treated with vehicle, with a solvent, towards the red line, which are normal controls. And you can see that the compounds highlighted here in dark green dots are compounds that achieve this. So we identified about 29 compounds that achieve this predefined threshold. So this is just a snapshot, and I showed you results from a library that included 1,000 compounds. But we were able to do the same thing for a library that was provided to us by a pharmaceutical company with 40,000 compounds. So we're able to scale up this assay to really include thousands of compounds and to ask in an unbiased way, can we identify a molecule that can help us change the molecular mechanism, that can help us restore normal cell function? And this is really the beginning of the pyramid. So we're currently at this stage where we identified a number of compounds through a primary screen, and we're doing confirmatory testing. And the next step really is to see if they not just work in skin cells, but also in nerve cells, not yet cell-derived neurons. We're working on this. We hope to end up with a list that we can triage and then further test in vivo. So that is the general approach that we take. And there are many, many steps that need to happen. But our goal is to have a compound that can be optimized for clinical use. And we're very realistic about the odds here. This is a note that I keep in my office just as a reminder that even if we identify a perfect molecule and we assume that we are correct in 80% of the time, the chances of this molecule going through all of the steps that I'm listing here is about 13%. So we're very realistic about the odds, but I think we have a shot, and I think we have a very strong basic science approach to this. So I'm pretty optimistic. And that is essentially what I wanted to show as to highlight both the challenges and the opportunities for this rare disorder. And I will say that although this is an ultra-rare disease, I think there is an important impact on other disorders, because if you can show that this approach works for one disease, that opens up opportunities for other disorders. And a lot of this work, or all of this work, would be impossible without the strong partnership with the patients that we care for and their families. This is from a recent conference we had at Boston Children's Hospital with a number, not so recent, I guess last year, with a number of families that joined us for the conference. We're very grateful for support, including from the Spastic Paraplegia Foundation, that make this research possible. We're also very, very grateful to the many collaborators, the contributors to the registry, and our many advisors, including Dr. Blackstone, who will speak later today, and Dr. Fink, and many others. So thank you for the opportunity to share our work, and I look forward to hearing your questions today and later. Thank you.